light-gathering organs all in a little crystalline array. The histology of the eyes is quite different. Single light-gathering lens for the mammalian eye and this array of independent little light-gathering units in the compound eye of insects. Although those eyes look very different, it turns out that the same toolkit gene is required for eye development in many different organisms. So this gene is called PAX6. If you make a mutation in one chromosome, one of the two copies of PAX6 during human development, you begin to lose particular parts of the eye, in particular the colored iris muscle that normally surrounds the pupil. So you can see there the eye of a patient with a defect in one copy of his PAX6 gene, and that produces the absence of an iris, a disease called aniridia, so the pupil now nearly fills the eye. Similarly, in the mouse, mutation in one of the two copies of the PAX6 gene partially reduces the size of the eye. Mutations in both copies completely eliminate the eye, the mouse embryo head shown at the upper right. Although Drosophila eyes look very different than mammalian eyes, there's a similar PAX6 gene in fruit flies, and if you make a mutation in that gene, you eliminate the compound eye of the fruit fly. Even more remarkably, if you overexpress this key developmental regulator of eye formation, you can generate new eye tissue in completely different body parts. So this is an experiment that was done by Walter Gehring's lab, taking this PAX6 gene and engineering it to be expressed during leg development in a developing fruit fly. You can see when you overexpress the eye regulator gene, you induce on the leg of the fruit fly a tiny little patch of red compound eye tissue. If you do a scanning electron micrograph, that structure has exactly the kind of independent repeated eye unit that you would find in the normal fly eye. And remarkably, you can get that result whether you do the experiment with the fly PAX6 gene or with a PAX6 gene from mice. So we think that genes like PAX6 are part of an ancient toolkit that's been inherited from a common ancestor and can be put to work to build related structures, even in very different looking animals. So how do these sorts of master regulators work? Well, many of the ancient toolkit genes that control the development of the AP body axis or the formation of a particular tissue turn out to encode gene products that act by switching other genes on and off. Okay, so for example, to build an eye, you have to express lens proteins and photoreceptors. What the PAX6 gene does is it acts as a regulatory molecule that flips switches on target genes and causes things like lens proteins and photoreceptors to be expressed at the site where the PAX6 gene is expressed. So we have a short video to show that kind of regulatory structure. So this is a gene with the yellow part of the DNA, the coding part colored yellow. That might encode something like a lens protein. It would normally only be expressed if an RNA polymerase lands on the gene's promoter and makes a messenger RNA from the gene. The coding region of the gene is surrounded by a series of regulatory switches. So these are parts of DNA that don't code for any protein. Instead, they act as switches that determine where and when the gene turns on. Those switches are the landing sites for regulatory molecules that bind to the switches, recruit DNA polymerase to the gene's promoter, and cause an increase in the total number of messenger RNA transcripts that's coming from the gene. Typically, a gene will be surrounded by multiple switches that allows the gene to be turned on at different times and places under the control of different signals and regulatory molecules. A lens protein in the mouse is also expressed in the liver, for example. It might have a switch where a regulatory molecule turns it on in the eye lens. The PAX6 gene is an example of a regulatory molecule that would bind to one of those switches. There'd be a different switch for turning the gene on in the liver. Okay, with that background on master regulators, let's come back to the problem of forelimb and hindlimb development in different animals. Forelimbs or hindlimbs are good examples of structures that vary in different ways along the anterior-posterior body axis. So forelimbs and hindlimbs can become wings or legs, short legs or hopping legs, spines and fins in fish. All vertebrates share Another set of toolkit master regulatory genes that are involved in controlling the formation of hind limbs or forelimbs. So these slides show a series of chick embryos that have been stained with a blue dye. This is a method that allows you to look at where an individual gene is being expressed. So you see sites of blue at the sites where the gene is normally turning on. There's a gene called TBX5 that's expressed in the wing bud of a chick, but not the leg. There's another gene called TBX4 that's expressed in the hind leg, but not the wing. There's another gene called PITX1 that turns on in the leg, but not the wing. So all vertebrates have these master regulatory genes that are expressed in one limb or the other and help determine how the limbs normally form. Doesn't matter whether you're a fish or a frog or a chick or a mouse. These genes are turning on in one limb structure or the other. So what's happened in organisms where the development of one limb has been drastically altered? So we went through yesterday the example of completely losing the hind limb in natural populations of sticklebacks. And I summarized a series of genetic experiments that showed that a major gene located at the distal end of linkage group 7 controls the presence or the absence of the pelvis in these natural populations. So it's possible to actually also determine the location of these toolkit master regulators that are known to be involved in hind limb or forelimb development. And when you do that, it turns out that one of the hind limb master regulators maps exactly to the locus that controls the presence or absence of the hind fin in the sticklebacks. That gene is called the PITX1 gene, the one I showed you on the earlier expression slides as well. This gene actually plays several different roles during normal development. You may wonder why it's called PITX1. Well, in addition to normally turning on in hind limbs but not forelimbs, it also turns on in some other body tissues. It turns on in the pituitary and plays an important role in controlling pituitary gene expression. It also turns on in jaws and mouth parts. If you completely eliminate the PITX1 gene in a mouse, you shrink the hind limbs, but the mouse dies at birth with pituitary abnormalities and jaw abnormalities, craniofacial defects. In some ways, that doesn't look very promising for trying to use this gene to evolve a new structure in populations that are subject to a full range of fitness constraints in the wild. On the other hand, if you look at what's happened to the PITX1 gene in sticklebacks, the protein coding region of the gene has not changed at all in marine and pelvic-reduced sticklebacks. In contrast, if you look at where this toolkit gene is normally expressed in embryos, there is an obvious difference in PITX1 expression. So now we're seeing blue at those sites where the PITX1 gene is expressed in either a marine or a pelvic-reduced population. You can see in the marine population expression in the blue mouth and jaw regions. It also turns on in a spot at the side of the body where the hind fin would normally grow out. You can also see that from the belly of the fish here in the lower left, with two spots where the hind limbs would normally form. In the pelvic-reduced population, you still have normal expression in the developing head region, the mouth, and the jaw parts, but at the site in the body where the hind limb would normally form, the PITX1 gene normally no longer turns on in that location. So what do we think is happening? That's summarized in the final brief animation. Both the marine and the pelvic-reduced population have a PITX1 locus. The coding region of the gene is still intact in both populations. This gene, we think, is surrounded by a series of these regulatory switches that cause it to turn on in specific body parts like the jaw or the pituitary or the hind limb. In both populations, some of those switches are still present and function normally. So the gene's expressed normally in the mouth parts. You still build normal mouths and jaws in both populations. Still expressed in the pituitary, you still form a normal pituitary in both organisms. In the marine population, it turns on in the hind limb and you build the pelvis. In the reduced population, the hind limb control switch has been inactivated. You no longer express the PITX1 gene at that location, and as a result, the hind limb fails to form in the fish. Okay, you'll hear from Sean that similar tweaking with these switches that surround genes provides a very flexible method for evolution to use to create major changes, morphological changes in particular body regions, but still preserve the overall viability, the coding region, and the expression of the genes in other tissues. Okay.